Welcome to The Microscopists, a bite-sized bio podcast. Hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on The Microscopists.
Hi, I'm Peter O'Toole, and today I spoke with a close friend, Lucy Collinson from the Francis Crick Institute, about the importance of failure.
Don't worry about the failures as well. You will fail as part of your career, and you will fail to stand up on a surfboard.
Speaking at conferences.
Yeah, I used to feel sick for a week before a talk.
And the Eurovision Song Contest.
And we wonder why we never get any votes at Eurovision.
All this in this episode of The Microscopists. Hi, I'm Peter O'Toole, and today I'm joined by Lucy Collinson from Francis Crick down in London. Lucy, hello.
Morning, Peter. How are you?
I'm good, thank you. I think the first time we met was ELMI, I think.
Yeah, ELMI in Leuven, which was my first ELMI meeting, and I have to say it's one of my favourites. It's a really good
I can see why you met me there.
Well, yeah, let's leave it at that.
No, let's not. Because that meeting for me was also very memorable for good and yes, all sorts of reasons. But I think the most obvious one is I think it was Sander who introduced us, was it, at the time?
Yeah.
And I was talking to Sandra about a potential grant application and thoughts about it, and he said, "Well, I know just the person who do this better than I could do it," which was yourself, and that's how we introduced. I believe.
Yeah, I remember you guys walking across the hallway in the middle of the university, very purposefully with your grant ideas, and it sounded really exciting. And me going, yeah, let's do it. And then 5 weeks later, we submitted and we got the grant funding, which is pretty much unheard of. Yeah,
and significant as well. I think it's my largest grant I've ever been awarded.
Mine too.
And I think with contributions, it was close to £2 million, I think.
Yeah.
That was it.
We bought a new scanning electron microscope with it. We turned it into a microscope within a microscope with Delmic, which is kind of cool, and started a lot of research for us on microscopes in microscopes.
Which, within 5 weeks. And actually, I think the backbone of the application was actually developed on the dance floor. Or around the bar of the conference banquet.
I mean, there wasn't much food, was there? But there was plenty of wine and plenty of beer. And I've never seen a group of people dance like the ELMI participants. It was excellent.
Yeah, I think that the most surprising thing is that we remembered the backbone of the grant application in the morning, because it was a long evening in the end. I was expecting it to finish much earlier, and it went on and on.
It might have been a different grant application as the one we came up with that day, but we got it. So
yeah, I think it was the simple ideas, and just, you know what, I think maybe it was a 5 week turnaround, but it was almost obvious.
Yeah,
and they're quite often the most successful, and the ones you don't have to think about too hard.
Yeah,
I just thought it fell into place. A lot of long hours putting it together, though.
Yeah, but that's not a bad thing when it's exciting, and when you know what you want to do and it fits with the grant call, and like you say, it just comes together.
So a lot of that was around correlative lighting electron microscopy, which is where you really forged your name, and obviously for 3D EM and volume electron microscopy. So that grant application was obviously a highlight, I think. What about the low? I think this is probably going to be similar. What have you found the most challenging times in your career?
The other grant that didn't get funded, the first big grant proposal that we put in, and it was, I love the acronym still, and I was umming and ahhing about whether to say the name of it, because I don't want anyone to nick it, but it's so good. So we called it iVoxel, which is imaging volumes with X-rays, electron and light. It's so good because it's all in 3D, so it's all voxels. And that was with Jemima Burden at UCL and Andy Bushby at Queen Mary. He runs the Nanovision Centre there. He's a material scientist. And Rafa Karzeniga, who's deputy head of my facility now, but she was at Imperial College. And the idea was to have a virtual network of volume electron microscopes. And that was pretty early on, before we even called them volume EMs. So serial block face SEM and focused ion beam SEM. And it would be open access so that people could send their samples in and run them. And it was probably about, I don't know, 12 years ago, something like that. And we didn't get it. And it was the first big failure. And I was so frustrated. And it, honestly, you know, when you see something and you can tell it's good and you know it's going to be beautiful and it doesn't get funded. And ironically, now we're just putting together the pretty much similar thing for a UK infrastructure for volume EM, however many years later, but the technology is much more mature and there's a much bigger community, and I don't know, it feels right. So fingers crossed.
Yeah, plug it quickly.
Yeah.
I know that whole community has moved on significantly. As you said at the start, volume EM wasn't really a word, a sentence. No.
Yeah.
Sentence. Phrase, maybe the better term to use it. But you've obviously forged your name in volume EM throughout that. But I've got to start that. So that didn't exist as a technique when you started electron microscopy. But when was your first contact with an electron microscope? When was that?
So that was the last year of my PhD. My PhD was in microbiology. And actually microbes aren't that interesting to look at, but at the time I was doing lots of biochemistry and isolating enzymes and running them on gels. And after 3 years of bans on gels, my boss recommended that I give my bacteria to the electron microscopy facility, which I did. And then you get a picture back and it was like, oh. Suddenly you can see what you're looking at. And I mean, it's not very, very interesting looking at a gum disease bacterium. There's not much inside it. But each of the postdocs I applied for contained electron microscopy. I'm not sure that it was, you know, I did it on purpose. It obviously caught my attention.
So what year was that?
98.
Ooh. Okay, so yeah, that was your first electron microscopy. You then went on to use electron microscopy more extensively straight after that?
Yeah, so I ended up doing a postdoc at UCL, at the Laboratory of Molecular Cell Biology in Colin Hopkins' lab. And I didn't really have the background for it. Oh, there we go, that's my leaving due. So that's around 2004?
2004? Yeah, yeah. So the picture we just put on,
yeah.
So I presume just above my head.
Yes. So just above your head is Colin, and Colin is a cell biologist, researches into membrane trafficking, and at the time he was quite unusual because he had his own light microscopes and his own electron microscopes in the lab. So I was taught and got to use the electron microscopes as fundamental parts of the project, but I also had to learn what a cell looked like because I had no idea, because I'd been looking at bacteria, so I didn't know what a nucleus was or mitochondria or endoplasmic reticulum or any of that good stuff. It was a very, very steep learning curve. But that photo is really interesting because there's a whole kind of, I guess, progeny of the lab that have gone out and run EM facilities around the world. So on your left, as I'm looking at you, Matt Russell is now a member of the EMSTP. And on your right, also Berninghausen runs a cryo-EM facility in Munich, and Jemima Burden runs the facility at the LMCB. So a lot of good electron microscopists came out of that lab.
And you haven't changed much at all.
Oh, you can tell the era by the hair colour. And the amount of time in lockdown by the grey hair.
Then through grey. I just keep cutting my grey off. Actually, I don't. I get my son to do it. Much better. Yeah, I wouldn't dare touch my own hair even. He's quite good. Yeah, nearly an end though. So hopefully.
Yeah, hopefully. Fingers crossed.
So that was your PhD. That was doing your postdoc days.
Yeah.
And then what was your next career step?
I then, so there weren't that many electron microscopists around, and you tended to find that you got lent out to other labs to do their electron microscopy for them as part of a collaboration. So after 3 or 4 years, instead of having my own postdoc projects, I was working on 3 or 4 different projects in collaboration with other labs. And I preferred that. I didn't really enjoy just working on one project and getting really deep into it. So I bumped into Dan Cutler in a lift back at UCL at the LMCB, where I'd started my postdoc. Colin's lab had then moved to Imperial. And he told me that the person who was running their facility was leaving. And I said, oh, well, that sounds like an interesting job. And a couple of months later, I took over running that facility, and it was just me and 2 electron microscopes and 26 groups. And only a couple of them were using EM when I started, and I think, I don't know, about 10 or 12 of them were using EM by the end. But I had to train everybody at that point because I was the only person there, so I couldn't do that amount of work. And that was a lot of fun, training people and working with people.
That's a lot of people. That's almost a third of the department, or more than a third of the department, using the electron microscope, which is pretty rare, I think, even today, to get such a large cohort as a percentage using the electron microscopes. Any idea of what it is at Crick at the moment?
Yeah, we have, so at full capacity we'll have around 100 research groups there, and at the moment I think it's about 95. We're working with around 60 of them. So it's not that people don't have a need for EM, but I think most of them are put off because it's not something they're taught to do hands-on, because the sample prep is really complicated, and ultramicrotomy, cutting ultra thin sections is very time consuming to learn, and you have to have a particular type of temperament, and you can't drink too much coffee.
Yeah, actually, yes. It's relatively early in the morning for you.
Oh yeah.
The one thing I was warned about very early on by yourself was in the first thing in the morning, not necessarily the early. So I've got a picture of your lab here, and they're all wearing hard hats, and they assure me they only wear that between 9 and 10 o'clock in the morning in case I bump into you. Is that true?
Yeah, it's only fair to warn people that I'm not on my best before 10 o'clock in the morning. I remember the only exception early on was if I was giving a talk, because I used to get really, really nervous about giving talks, and I would panic for about a week beforehand. So I would ask to be on first just so I could get it out of the way. And my first big talk was at the Society of Electron Microscope Technology in London, and I was on at 9. So it was a very fine balance between, not being with it because it was too early in the morning for me, pre 10 o'clock, and getting the talk over and done with. And then the projector broke down on me, so it kept overheating. It would turn itself off every 45 seconds. And I was talking about some work we'd done with correlative light and volume EM on zebrafish, looking at blood vessels growing. So I had to mime anastomosis, which is blood vessels growing and then fusing. So, maybe it's good if it's early and you don't have too much time to think about it.
So you're a lot more chill now. Which, yeah. If you caught any of your staff with their boots on one of the EM tables, you would go nuts.
Absolutely.
I violated your warranty as well, by the way, sorry.
No, absolutely. And I knew I shouldn't have been doing that, but that is a look of absolute relief, because that was the first microscopes on site at the Crick after 8 years of planning and 5 years of building. And those microscopes had been on the back of lorries across London and into a new site. So I wiped it down afterwards.
It's an exciting time, though, to take up the new role at Crick and move through. Have you ever regretted moving out of being a pure academic and that type of route and taking more of a core facilities and leading the core facility there?
No, never. It wasn't for me the traditional route through science. I didn't have my own research question, but when I started using electron microscopes and light microscopes, it was the technology that really interested me and excited me, and it was pushing the technology forward to be able to answer the biology that really motivates me. I guess one of the difficult things that you'll have experienced as well is because we're pretty much the same age. So when we started in core facilities, it was a non-traditional career, and people tended to think of you as a failed scientist. And actually, looking back, it's changed. We've had some very dynamic people going into core facilities and really building them up and driving it as a real career. And as technology gets more complicated, so you need people who have a background in biology or materials or physics or whatever to make these things run for particular applications. And it has changed quickly, and I think now a lot of the researchers that we collaborate with see us as, understand that we're scientists with similar backgrounds to them. We've just gone off in a different direction, but it's not completely plain sailing, I think.
Yeah, you're right. Our posts are clearly not academic in the normal term of academic posts. We don't lead research groups, purely research groups. But I think it's fair to say that we have quite a large influence over the research going on at the institutes, universities, and even our own teams are academically minded in some cases and driven. I think you're probably one of the leading examples of how in a non-academic post you are very academic still.
Thank you.
and I think there's a delicate balancing act from having your own research interests and also which is technology advancement more than answering a biological question, because I think it's fair to say, similar to myself, you're a technologist interested in advancing the technology for the users, and then also keeping that balance that our teams are still service orientated. I think that's important.
But I'm allergic to the word service. As soon as you say service, people think that they can just bring you a sample and give it to you and say, I want a pretty picture for the end of my paper. And actually what we do is, you have to collaborate. They have to put the effort in and tell you the whole story behind their research and what they want to see, and therefore what resolution you need and which probes you need to get into the samples and how you're going to prepare them. And there are as many different correlative workflows as there are research projects. So everything is custom designed, the research.
Yeah, I think I know from some of our side, I do see us as a service at York for some instances where it's pretty routine, turn handle, get results out, but not in all cases. Clearly not in all cases. And they're the ones, I think the team and myself enjoy both sides of that. I don't think service is a dirty word. I think we're still serving it. We're still part of their team. We're still adding value. We're still going to be inputting into their research, but at a different level. I think it's that extra level then, taking it beyond what they've come and asked for. And that's certainly what you've been doing in a significant way. And I think citizen science is just one example of that, of how you've got a problem for users and you've taken those problems and moved it forward. You found a solution. So I think this backdrop is from what, Lucy? So I've got a very icy picture here.
Yeah, that's Chicago last year in March. It's really cold in Chicago in March. We just missed the deep freeze, but that's looking out from the meeting room at the Adler Planetarium over the lake, which was completely frozen. And the reason we have a citizen science project is because of microscopes like this. This is our focused
That's your Gemini 2?
It's the 540, Crossbeam 540, which is the focused ion beam scanning electron microscope. So we have our cells and tissues embedded in resin. We use the ion beam to mill away very thin slices, 5 or 10 nanometers, and then you take an image, and then you mill an image, and we can collect thousands of images in a week. And milling at that scale even to image through an entire cell, just one entire cell would be, say, 3,000 to 5,000 slices. So then your data is up in the terabyte regime, and we just can't analyze the data quickly enough. So you can't extract all the meaning from it. A 2D slice doesn't tell you very much, but if you can convert that into a 3D model of the cell with all of the organelles, then you can start to be quantitative and compare between health and disease. So we were a bit stuck, and we have the EM side of the team who are working on research projects, and then we have the microscopy prototyping side run by Martin Jones, who's a physicist, and Martin and his team build new hardware and software. So it was actually Martin's idea to go to citizen science, because now we're in the era of machine learning.
I'll just
Martin.
grab Martin, picture of.
I'll tell you what he's holding in a second. So now that we're in the era of machine learning, what you really need to train computers to recognize organelles automatically is ground truth, which is normally means us experts sitting in front of a computer and drawing around, say, the nuclear envelope in thousands of slices.
So it takes a week to get the data set.
Yeah, in Fib, yeah.
And then how long for that expert to analyze that data, to draw around it and make a 3D volume of it?
So just for the nuclear envelope, which is the membrane surrounding the nucleus.
Lovely images, that.
And that's also what Martin's holding, that's lit up in red. That would take another week. And that's just one structure inside the cell. That's, not even thinking about the other organelles like mitochondria, endoplasmic reticulum, Golgi, etc., etc.
I'm still impressed it's only a week to actually do that.
Yeah. Well, guys in the lab are really good, but that's one cell, one week working on nothing else, and you're very long.
Statistically, not very robust.
Yeah, statistically insignificant. So we, through Martin's original idea, we collaborated first with CR UK and then with the Zooniverse, which was set up by Chris Lintott in Oxford, and he uses citizens or the general public to help him classify galaxies. And his project went, so this is back in about 2008, and his project went so well that Galaxy Zoo, his original project, became the Zooniverse, and the Zooniverse now hosts a huge number of different citizen science projects, and we had one of the first biomedical projects asking the general public to do this drawing and to reconstruct organelles for us. So this one and the one that Martin's holding is the first nucleus that was completely reconstructed by the general public.
Can you trust the general public to draw and annotate it properly?
Well, I always said no. I always said you had to have 10 years experience looking at EM images to be able to understand them and interpret them. And I was completely wrong. You've got that audio recording of me saying I was completely wrong now.
Brilliant.
It turns out that you give them a few short instructions, and people do a pretty good job. We get some graffiti, some of which we can show and some of which we can't. But Martin and Helen Spears, who's from the Zooniverse, who's seconded to us, who's their biomedical lead, and our digital development team at the Crick, are able to automatically remove the graffiti. They've designed algorithms to pick out the right line from all of the different citizen science annotations, and then train the first machine learning algorithms, so the paper's almost ready to bioarchive, which is really exciting.
That's your latest research then?
Yeah.
How many citizens? So just Joe Public, these aren't scientists necessarily, these are hired people in many cases.
Yeah, you get to know, so there are chat boards on the site, so you get to know some of your citizen scientists. And early on, one of the people who went through the entire data set that we put up really quickly was a grandmother in the US who'd broken her ankle, and so she didn't have anything else to do, so she did all of the images and then asked for some more. And wow. We have schools doing it. I've just been asked by a couple of people for references for medical school for their contributions to the project. So, and it's building. So we now have a mitochondria project out. We're working on an ER project, and then we'll hopefully start putting up some really kind of cutting edge crick research on there, and pushing forward into virtual reality as well, because at some point we're going to have to visualize these things and turn them around and start to understand their 3D shape and structure and what that means in biological terms.
So how many volunteers have inputted so far?
It's on the first nuclear envelope project, it was around 5,000, and we had around 150,000 lines drawn. That's actually quite a low number for the Zooniverse. So they have about, I think, 1.5 million to 2 million volunteers who interact with their platform. But a lot of those projects are saying yes or no to questions, whereas ours, people actually have to sit down and really interact with the data. So there are fewer people.
Yeah, I think maybe as word gets out and it passes around more, I think the importance of what they're doing is huge. I mean, this is them volunteering and helping solve very difficult questions, which ultimately aids cancer research significantly.
Yep.
I guess that gives them a sense of reward. But just looking at your image, which looks like a load of fabric web, so I guess it is virtual knitting in a way.
Well, maybe that's the next public engagement project. Maybe we'll get somebody to crochet a nucleus. We can have a whole, I think they did that up in Scotland, one of the institutes I visited. I think they knitted a cell or something, which was in the middle of their atrium.
Wow. So this is going to be published soon?
Yes.
Well,
bioarchived.
I'm now going to ask you a difficult question. And I'll allow you two if you can't think of one, but what is your favourite or most favourite publications that you have been part of? Difficult question.
It's usually the most recent one.
Okay.
Every paper has a huge amount of effort from the guys in the lab. So our most recent one is with Eva Frickel and Serge Mustavi from Birmingham and Imperial College, looking at toxoplasma parasites in zebrafish brain, using zebrafish as a new model to study the infection. And Mary Charlotte in the lab has just done some incredible correlative light and EM in 3D. And we're lucky we have a whole range of microscopes, so we can pick and choose them as we need for the biology. So she's used, this is real needle in the haystack thing, so it's whole zebrafish brain with these tiny parasites in. So she's used X-ray imaging to locate them. There's really beautiful fluorescence imaging of the parasites and macrophages. And then we have serial section TEM, focused ion beam SEM, and serial block face SEM of around 40, something like 40 different parasites. So it's I guess the beginning of our volume EM work was back looking at blood vessels and angiogenesis with Holger Gerhardt's lab, again in zebrafish, and that was the coarsest correlation, but it was all we could do at that point. And the most recent zebrafish paper is, I think it shows how far we've come in the last 10 years or so.
So where was that published?
That's a really good question. It's on bioRxiv, but it's just been accepted. I need to double check the journal.
I'm giving you a plug. The opportunity to plug your paper, increase that factor.
I know. You can tell that it's, I'm really thinking about this as you're asking me. It's not pre-prepared.
I think that also shows that it's not about where it's published. It's about the fact that it is published and out there for the public to read that is more important.
Yeah. In some ways it does matter, right? It matters for metrics and for careers, but it also, I think, we don't have the right metrics to measure non-traditional careers yet. And the other aspect is that, the Crick is able to make everything that it publishes open access, which is fantastic, but really there should be a big push towards everything being open access.
Which I think there is certainly
Yeah.
across Europe,
Yeah.
UK. I think collectively, I think it's not just a UK thing, it's a very European initiative towards that. And actually, I'm also aware that you're also quite passionate about Europe in general.
I know what's coming.
I'm struggling to find the image. Drat. There you go.
Oh.
Yeah, so probably not the one you were expecting to see.
No, but I love that. So I feel like, so that's
what does it say on the poster?
It says, and we wonder why we never get any votes at Eurovision.
Yeah, I think that sums you up twice over. There's two important messages in this.
Yeah. I mean, I have a banner. I feel like I've spent the last 3 years going on anti-Brexit marches. And I have a nice banner in my office as well saying science needs EU, science needs you. But of course, yeah, we do love the Eurovision. I mean, it's kind of a British tradition that you have a cheesy party for Eurovision where everybody brings food that is a pun on the different countries that it comes from.
I wouldn't say that's everyone, Lucy. I would say you are. I wouldn't say. I've got to be careful how I word this.
Yeah, be careful.
Yes, you are. You are a Eurovision fanatic. I would argue at this point.
Well, it doesn't hurt that it's around my birthday every year.
So this was a memorable birthday, I think.
Yeah, that was a big birthday. And the year before, we had our usual Eurovision party, and somebody said, well, because it's your big birthday next year, then we should actually go to Eurovision, wherever it ends up being. And it was a close call between Copenhagen and Azerbaijan. And we ended up in Copenhagen. And it's a great way to see European cities with a bit of cheesy music.
And am I correct in that you have a Terry Wogan dress?
Well, you might think that from our Twitter feed. So in lockdown, we've had lab coffees, and we have a different theme every day. So of course, for my birthday and Eurovision, we had Eurovision theme. And I didn't have a green screen, and my background wasn't working, so I had to hang a green dress in the background, and Terry Wogan's face was on it, because for us in the UK, Terry is Eurovision. all hail Sir Terry.
Oh, Graham Norton's not so bad.
Oh, Graham's great as well, but I grew up in the era of Terry.
So that's important, actually. So your team obviously mean a lot to you. Obviously, we saw them with hard hats earlier. But actually, I looked at this photo. I think the photo I have now is of your lab on the last day before lockdown.
It was the last kind of big lab training event. That's, they're all having so much fun there. So this was in preparation for the Zeiss 3D EM-LM meeting that we were holding at the Crick, supposed to hold at the Crick in March. And we had to take the difficult decision to cancel it or postpone it. And it was before we really knew whether we were going to have to go into lockdown as well. But Zeiss had shipped an SEM on site, and they were all learning how to use STEM detectors and use the array tomography software, and they're having so much fun. Kind of makes me a bit sad that I wasn't there. I don't get on a microscope very much anymore. No. But they're such a talented group of guys. If I can't get on a microscope, then the fact that every week we catch up and they show me this insanely cool data and reconstructions and analysis it makes me happy.
Which is good. And obviously this isn't your whole lab. Your lab is actually, this cuts people out at the edges, obviously a lot larger. If you think about your postdoc lab, and Colin Hopkins was a great scientist, look at the lab that you had here, the size of your own lab is significant.
It is now. Yeah, it is now. But
yeah.
I get in 2004, it was just me, and it's been a very gradual build up over the years. As the size of the institute has grown, you need more people, and as the technology develops and as you show what it can do, then more people want to use it. So
I think, I have a picture of you, and I haven't got it downloaded for some reason, which is frustrating because you'll know what it was going to be of. I think I started similarly. There was myself and there was Meg, who's my electron microscopy specialist, and the lab grew and grew and grew. And I think we were both very fortunate that our timing of coming into core facilities is when core facilities started to come into being, but also the technologies that we were involved with were very much in vogue and in fashion, and were very successful to develop. But to develop the lab, you really had to, it is a good analogy. It is like surfing, and you have to surf the wave successfully to get there. And I don't have the picture, but I noticed behind you is probably from one of your holidays, Newquay or similar.
Polzeath.
Okay, because
Polzeath
you're
the north coast of Cornwall. I don't know if I can pull up the photo.
So out of work itself, it's a big team to manage. I'm sure lockdown. Oh, look at that.
That's the other side of me.
Can we go sideways on it and look at it?
I'm not sure why I'm on the side, although I have to say when I surf, I love surfing and I talk about surfing a lot, and every holiday is a surfing holiday. But that gives the false impression that I can surf, and I can't. I didn't start till my early 30s, and I've never been sporty. And it's not about that at all. It's just about the feeling of being in the sea. And when you're getting slapped around the face from both sides by a wave, and I don't even know how that's possible, but you can't think about work. And even if there's no waves, you can float in the middle of the sea, and you've just got the sun and the air. The air smells a lot better than it does in London, and it's great. And look at the view. It's so beautiful.
And the salty water and the cold and I'm not getting it at all. I'm just
Yeah, I mean, I didn't like the sea before I started surfing, and there was no way that anyone was getting me in a wetsuit, because just look at them. And then one day I tried it, and the first day you have a lesson, most people make some attempt at standing up, and most people get hooked. So we have lots of group holidays down to the north coast of Cornwall, and actually there's quite a high proportion of electron microscopists on those holidays, I have to say.
not surfing.
Yeah.
Okay.
Go figure. It's because we're locked in basements most of the year in the dark, so every now and again we get let out and we go crazy.
You'll get wiped out instead of blacked out, I guess, on the microscope. So back to so you have successfully surfed that wave, career-wise, but how have you found actually challenging? The first picture I showed, you mentioned the Terry Wogan dress, but you've kept your lab fairly well engaged throughout lockdown. And I remember on Twitter, actually, from Martin, I think it was Martin Jones who actually tweeted this. This was a very scary image of you, I think.
Okay, so this one, so the idea for themes.
So you're not really home alone, though.
No, this one was the Scream. I think it was probably like week 3 or week 4 of lockdown, and everybody was having a real dip. So half of the lab had the Scream, the painting, as a background, and half of us had the Home Alone, and we were doing the Scream. And then an unnamed person in the lab took that and ran with it, literally. And I think that unnamed person is probably Martin. But there's now a whole Twitter feed called #Lucy running away from things.
But there's all sorts of backgrounds. I think ultimately you enjoy virtually dressing up.
Yeah, I mean, I love it. It makes me laugh, and I think if there's anything we need right now, it's having a good laugh. And
I think
now and again they come up. I've been chased by the ghost from Ghostbusters and Teletubbies and Giant Haystacks, the wrestler, and all sorts. So whoever it is, I encourage them to continue.
Yeah, I think good for team morale as well.
Yeah.
So you've obviously been doing a good job from that perspective. There was one picture that you sent me, which I was very intrigued by, which is actually a picture of an aeroplane and the distance of you on the aeroplane to McDonald's for chicken nuggets.
Yeah, you might have to go the other way so that...
There's a plane.
Okay, yeah, so there's me on the aeroplane. So my brother loves aeroplanes, and he follows my travels around the world on flight radar every now and again. Not the whole time, that would be weird, right? But he knows which.
Would you travel a lot?
I know. He knows which plane I'm flying on and whether it's on time, and it got to the point where he was texting me just before landing. So when I turned my phone back on, I'd have a message saying welcome to wherever. So that one is Frankfurt, and I think I'd arrived quite late, and he thought I might be hungry, so he's pointed out the nearest chicken nuggets in McDonald's, which I think was the only restaurant labelled on there, to be fair.
So you're not a big McDonald's fan?
Do you know that I have been known to have a McDonald's in Frankfurt Airport on the way home?
Yeah, I think I've done similar. They've also they've got Haagen-Dazs as well in there, so I kind of actually their waffles there are quite good. So that's usually where it's moved. Last year it's moved, so I no longer get that when I go there.
The irony being we're both quite foodie, aren't we? So I like that we're talking about McDonald's.
Yeah, I'm not so fussy though. I think I do enjoy McDonald's occasionally, too often.
The right amount.
So, God, some quick fire questions then, and we're going to start with food. Eat in or eat out?
Ooh, depends on the day. If it's the weekend, eat out during the week. Cook at home.
Great quick fire answer, that one, Lucy.
What's quick?
Okay, cook or wash up?
Cook.
Cook. And have you ever told Chris that? So we have a friend called Chris Guerin, a common friend who we both know very well, and when he ever visits, he'll always cook. That's his big thing, is cooking. I
mean, he's been a chef. His cooking is insanely good, and he came down to Polzeth with us last November. We took him on a nice beach holiday, and we actually stayed on a place on the beach, and it was gale force winds for 4 days, the poor guy, and but he cooked us some incredible food. So always try and take Chris with you on holiday.
But you still rather cook than wash up when he's there?
No, I'd rather he cooks. I'll wash up.
Because he will be listening to this. You have to be careful. Okay, beach bum or sightseeing? Quick answer.
Well, I'm going to have to say beach bum because of the surfing.
Okay, tidy or messy?
Messy.
Tea or coffee?
Coffee.
Book or TV?
TV.
Okay. Stay at home or go to work?
Oh, I don't have much choice at the moment. I go to work. But if you'd asked me when I was at work every day, I would have said stayed at home. Grass is always
how's Ian going to take that then? Your partner back at home. That you now say you prefer to be at work.
Yeah, I think he quite liked me out of the house now. It's been, 3 months?
So you miss him when you're at work, and when you're at home, you obviously want away.
Yeah.
Get yourself into trouble with that. So TV. So you prefer TV over books? I would concur with that. But so what are you into at the moment, TV wise?
Well, my favourite is Spooks, which, if anyone hasn't seen it, was a series about MI5, not 9 to 5, and it was kind of, I guess it was around the time that 24 was on as well, so it was very British, but it was the first one that had that really tech feel. That would be my go-to. I am trying to read, but it's hard to concentrate, I think, with work and with Twitter and with everything being so quick. And is this still the quickfire round? Because I'm
no, don't worry. I've got you into trouble with the other half, so I'm quite happy to stop there. Yeah, and if you've got young children, don't watch Spooks with them.
No, especially not the first episode.
Correct. Yes.
Yeah. Should come with a warning.
Yes. I remember that, that's one of the few vivid moments of TV I remember, watching that scene and thinking, oh my goodness, 'cause it was a big actress as well.
Yeah.
And
well, they were one of the first that I remember that quite happily killed off major characters.
Yes.
And did it frequently.
Yeah. Was that first episode really on a Friday? Like, it was a good pun in there for anyone who's watched that first episode of Spooks.
And I have to say that, because we've had a couple of master's students from your course in York, and both of them have been really excellent punners. So, you know, you're not only teaching them microscopy, but you're also making sure that they go out into the world able to pun.
That's Catherine and Charlotte, I think, both came down to you, didn't they? And
Yeah.
Catherine's now working for you as well.
Yeah, Catherine went over to Graham Knott's lab in Switzerland, and then we managed to get her back, and she's fantastic. She's working on projects and helping Rafa with operations. And Charlotte is now in Oxford, so she did a PhD in Leiden. So it's one of the really nice things about science that you don't get until you're a bit, I don't think I can say older, can I? Maybe later career, when you start seeing your kind of lab kids go off and do their thing and then build their careers and come back again, and that's very cool.
Yeah, I think maybe that is not common of all core facilities. That we have had students come in and then carry on their career around the technology rather than the typical academic route. And both of those students were master's students at the time, and they're meant to be trained to go into our type of roles or similar in industry. And I think masters is not always commonly thought of as a good degree compared to a PhD, but actually it equips them really, really well. And in Charlotte's case, she's obviously gone on beyond that anyway, but they can still forge very high profile careers. And specialize in that technology. It's a very different route, but still needs that high degree of expertise and specialisms, just as you would in an academic career. I think you've got two excellent examples there. But you helped inspire them significantly once they went down. I think that was the London Research Institute at the time that you were at.
Yeah, it was Cancer Research UK, and they were both paired up with the postdocs in the lab with Chris and others, so they were learning directly hands-on from some really great electron microscopists.
Which is fantastic. I'm going to just turning back to science itself, and we've talked about where you started, where you've got to. What's the greatest challenges that lies ahead? Where's the unmet need that we need to see developed? What's going to really drive microscopy forward in your area in the future that isn't possible today?
For the automated image analysis. So that's where we're devoting a lot of our development time now. It's partly the citizen science, it's partly deep learning. A lot of it is, I guess, things like NuBIAS, which is a European network of image analysts building up the career structures and producing more people to come and work in facilities and in research groups to develop it. Places like EMBL EBI, the European Bioinformatics Institute, who have the public archives where we're starting to deposit our data so that image analysts and other scientists can remine it. That goes to open science again. I think there's a huge amount that needs to be done in that area to really equip everybody to be able to collect your big data, whatever it is, whether it's volume EM or light sheet or spatial omics or X-ray microscopy. You collect it in 24 hours, and 24 hours later you have whatever you need to quantify. In volume EM, that's going to be 3D models of organelles or of neurons, and some way to quantify them. In other domains it might include time as well, and high throughput, which needs automation. So EM has never been high throughput, but you're starting to see kind of the first big data coming through fast, and places like EMBL and Yannick Schwab's lab are really pushing that kind of high throughput work that will make the data significant as well.
Which I was thinking when you said about the analysis and the high throughput analysis, as soon as that comes to be, we then need the high throughput of the data. Yeah. It's where the bottlenecks exist, I guess, at the moment. Does it not frustrate you that deep learning and computers cannot easily render our images?
It doesn't frustrate me. I think if they could, we'd have to worry a bit, because we might have reached the singularity. So it doesn't frustrate me. I think it's really exciting that there's the potential that we can teach computers to do some of that heavy lifting for us. And actually, we've had 2 sandwich students in a row, Harry and Daniel, who come from a computing background, and these are undergraduates, and these are the guys who are really doing some heavy lifting on the machine learning side. And it's just a new generation with different expertise, and when you get them working across disciplines with biologists and microscopists, it's really exciting.
Yeah, I've got someone from maths, compute science, working as a PhD in the lab at the moment, and I guess my frustration is I don't know the work as well as they know the work.
Yeah.
And so it's hard for me to understand their bottlenecks, their problems they're encountering, because conceptually it's really easy.
Yeah.
But obviously it's not, because no one's solved it.
No.
And I guess I feel out of my depth or inadequate in my knowledge base. I can't advise them particularly well. We've got obviously another supervisor who's expert in that area. And I think that side frustrates me, and maybe more frustration of myself, that I expect great things and I think it should be easy to solve, and I appreciate it isn't. But it should be. Why isn't it?
You need a translator. And as well as, running the research programs in microscopy prototyping, Martin is basically my translator for hardware, software design, engineering, machine learning. And it's fiendishly difficult. It can take years to not learn another topic, but learn how to communicate with somebody on that other topic. And he's extremely patient. He's the person who's got me Twitter enabled, and I even know what a Jupyter notebook is now. And I've done a kind of a 0 to hero Python training, which is a fantastic course if anybody wants to do it. But I'm never going to be an expert in coding. You just need to know enough that you can communicate, and you all need to, bring your expertise to bear to get to what you need for the research.
I think that's very good advice, actually, and I would completely agree. And actually, my students and Julie Wilson, who I collaborate over in maths, are very patient with ourselves, and they do explain things very clearly. It's just when I leave the meeting, I lose some of that information. I think they have to keep reteaching me quite often, because it's just not instinctive to me at all.
Well, it's very different. Somebody explaining something simply to you and you understanding it, that's very different to being able to then go and explain it to somebody else in the same way. I
think the tolerance goes both ways, because we have to explain what is quite simple to us, but as they're not biologists, not microscopists hardcore, again, I think it goes both ways. Yeah,
and biology is messy, right? Much messier than physics and maths, and that's, I think, the thing that people from the other side have to get to grips with.
Yeah, and chemistry also. I guess if you add A to B, you get C.
Yeah.
Quite often, and it's repeatable.
Yeah.
Whereas biology, the heterogeneity means that you'll never get the same result twice.
Yeah.
Trends, but not the same. Well, think about your nuclei. They're never going to look the same, are they? Each one is going to be completely unique, even if it's from the same cell type. In fact, the same cell, seconds later, will be different.
Yeah. You need a lot of data to start spotting the patterns.
That heterogeneity does make biology very, very difficult, and why it's so expensive.
Yeah. But it's a problem that's been overcome largely in the omics. It's just that imaging isn't there yet.
No, but I guess it's the omics and coupling to that's a big route forward as well in the future. Yeah. So before we finish off, I'd like to ask you about what's been the best time of your career? What period of your career have you most enjoyed?
Good question. I think now, because
this very second, because you're loving the interview so much.
Right now, you're doing such a good job, Pete. No, I think for a long time it's a struggle, because everybody seems to know everything, and you don't know everything, and you think that you should know everything, and it's only once you've done it for 20 years or more, and you've collaborated with people, and you've picked up stuff from different subject areas, and it's only when you learn all of the stuff, especially in biology, that it starts to all come together, and you suddenly realize that you're there, and you know what's missing in your field, and you know what you want to do with it, and you can have conversations with different people in biology or in maths or in computing or in physics, and I think for me the fun bit is when you see the data that the guys in the lab are collecting, but when you have that network and you can start putting together really big scale ideas that could really make a difference to science on a much bigger level.
So I guess all that comes with confidence.
Yeah, which takes a long time to build up.
Now, how confident are you now when you invited to give a keynote, a plenary talk at a big conference? Do you still get nervous, or are you now a lot more relaxed and confident with it?
A little bit, but I used to feel sick for a week before a talk when I was doing my PhD and my postdoc, and for a long time, and now it's one of the things that I enjoy the most, actually. And I think you just have to keep doing stuff. If it makes you nervous or you don't enjoy it, you just have to keep doing it, and eventually you get better at it, and then you get a bit of confidence.
I guess a lot of people always worry there's people in the audience that know more than you know about the subject, or might ask a difficult question. But I don't think I've ever encountered a really, I'm going to set myself up now for the next time I give a webinar or conference. I don't think I've ever had a really difficult question that I should have known the answer to, but didn't know the answer to. Because I think it's fine not to know the answer for a question.
Yeah.
And just to say, I'm not the expert there, and ask the audience or come back and I'll find the answer out.
Yeah. It's okay not to know, as long as you say you don't know. Do you only get into trouble if you start to wing it and make it up? Because there will be somebody in the audience who does know. You can bet on that.
Yeah, no, better in a very specific area, but probably for the whole content of the talk, probably not.
Yeah.
I think
it's like when you do your Viva, your thesis Viva, right? You're the person who knows that work best.
So, again, just some slight bits, some of the pros, some of the perks, maybe, of the job. So you sent me through this picture, which is of you talking to Sadiq Khan, is it?
Yep.
The Mayor of London.
Yep.
A very influential
person. He's just about to take part in our citizen science project. So the screen on the left, just above your head, has Etchacell on it. That was the beginning of London Tech Week. And he was fantastic. He didn't have long at the Crick, but he came over to the screen and he segmented an entire nuclear envelope in one of the images.
So is he co-authored or acknowledged on the publication when it comes out?
Well, we have our volunteers as co-authors. So there's, I think they're called Zooniverse volunteers in the author list, and then it will point towards a list of everybody who's contributed on the project. So it's really important to acknowledge everybody. Again, it's very non-traditional science.
So he's going to be acknowledged. You're going to have Sadiq Khan as a co-author.
Not specifically. He should be in the list with all of the other volunteers. I'm sure you wouldn't want to pick him out.
About this one, which is now you with the Queen. Tell me you got her to do it, and you can co-author. You can put her in the list as well. Go on.
We didn't know. No, that was before the Citizen Science Project launched, I think. So that was the official opening of the Crick, and my name got drawn out of the hat to be one of the people who met her in person and introduced her to other people. And I just love that we've coordinated our dresses there.
Now, I was thinking, so you met Sadiq Khan, you've met the Queen. I think you gave a big New Scientist talk, I believe, well.
Yeah.
So you get to do all these gigs. So that means you get to buy dresses. How often?
Well, like shopping.
but not at the moment, obviously.
At the moment, no. I mean, this is the most dressed up I've been for 3 months. I have to be honest.
except for virtually, in which point you've been dressed up in all sorts of different outfits.
Yeah.
That's the virtual side. I say we're coming towards the end now, so I'm going to ask one last question. Any advice that you would give to someone starting out in their career?
Another good question. I think follow what you enjoy doing. I mean, nobody in primary school says I want to be an electron microscopist, do they? So
I hope not.
I mean, maybe they do now, right? Then maybe they know about it because of
It's in the
ETCHSL.
syllabus, certainly for A levels.
Yeah. And we've done a bit of teaching of teachers, but I think do what you find really interesting and you're passionate about, and it may be that you only know a little bit of that. So at school I liked biology, so I did a biology degree, and then I liked the biology degree, and I quite liked small things, so I did microbiology for my PhD, and there wasn't a grand plan, and I didn't know where things were going to lead. You have to develop a gut feeling for which opportunities you should take and who are good people to collaborate with. So, and don't feel like you should know everything too early on. You have to spend a lot of time in science learning before you get to the point where you can really know what you want to do.
I think that's sound advice. It is just following what you're good at and the opportunities. Again, back to surfing, I guess.
Yeah.
Which you might get back to sooner than later.
Yeah, don't worry about the failures as well. You will fail as part of your career, and you will fail to stand up on a surfboard, but it doesn't mean that, you learn from it. This is very deep, isn't it?
Yeah.
You do learn from it, and I think it makes you a bit more chilled.
Yeah, okay. I'm going to caveat what you just said there before we end, because if your true surfing skills are what that you say they are, i.e. pretty useless, career-wise, it might be better to find a different hobby. From a career perspective, fall off. If you keep falling off the same surfboard, maybe it's time to choose a different career path.
That's why we only surf places that are shallow with a sandy bottom.
And on that note, Lucy, thank you so much for joining me today. It's been, I would say, enlightening, but that would suggest light, and it's been electronifying. Maybe would be a better fun to end on. I'm
going to put that phrase in my next grant application.
Cool. Lucy, thank you very much.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.